Hi, welcome to Revision Chapter 1, Macromolecules. This is what we have learned so far in Chapter 1. In introduction, you were introduced to chemical bonds, starting from the strongest bond to the weakest bond. And then you are introduced to functional groups such as carboxyl, carbonyl, amino groups, and others. And then you are also introduced to isomers. Next, we move on to properties of water. In properties of water, you learn about high specific heat capacity, high heat of vaporization, water as good solvent, high density of water, transparency of water, the activity of water, and also the cohesion and adhesion in water. After that, you are introduced to macromolecules. There are 4 macromolecules introduced to you, where 3 of them are polymers and 1 of them is not a polymer. And you will also learn what is polymer and why does lipid considered as not a polymer. The first macromolecules introduced to you is protein. In protein, you learn about the monomer of protein, which is amino acid, the groups of amino acid, polymer for amino acid, which is protein, and bonds that bind the monomer together, and levels of organization, which we have primary, secondary, tertiary, and quaternary, and classification of proteins. After you learn about proteins, you learn about carbohydrate. In carbohydrate, you were introduced to 4 types of carbohydrates, that is monosaccharide, disaccharide, oligosaccharide, and polysaccharide. Monosaccharide is the monomer of carbohydrates, whereby disaccharide is the combination of 2 combination of 2 monosaccharide. Oligosaccharide is the combination of 3 to 14 monomer of carbohydrate, and polysaccharide is the combination of more than what we have in oligosaccharide. Monosaccharide can be classified into 2 ways. One is by the number of carbon, the other one is by the location of the functional group. Triose sugar is a 3-carbon monosaccharide, and pentose sugar is 5-carbon monosaccharide, and hexose sugar is 6-carbon monosaccharide. Example of hexose sugar is glucose, fructose, and galactose. Disaccharide is the combination of 2 monosaccharide. Combination of glucose and glucose will form maltose. Combination of fructose and glucose will form sucrose, and combination of galactose and glucose will form lactose. There are different bonds that hold disaccharide. Generally, carbohydrate is held by glycosidic linkage. However, the number of the glycosidic linkage will show the the number of carbon that bind the 2 monosaccharide. In polysaccharide, it is easy for us to divide it into its function. The first function of polysaccharide is as storage, and the other function is as structural support. So Starch is a storage, carbohydrate storage in plant, and glycogen is carbohydrate storage in animal. For structural support, we have three types of polysaccharide. One is cellulose that is used as the cell wall of plant, chitin, and we also have murate. After learning about carbohydrate, you are introduced to lipid, and lipid is not a polymer because it is not formed by the combination of monomer. There is no repeating unit forming lipid. Three types of lipid introduced to you. One is triglyceride. Next one is phospholipid and steroid. Triglyceride is the combination of glycerol, one glycerol, and 3 fatty acid. And it is held by ester linkage. Phospholipid, on the other hand, also contain glycerol, but the glycerol is binding to phosphate and choline, and the other 2 carbon is binding to fatty acid. So for phospholipid, we have hydrophilic head and hydrophobic tail. Next one, you are introduced to steroid that is easily identified by the 4 fused ring. Example of steroid is cholesterol. Finally, you learn about nucleic acid, and the monomer of nucleic acid consists of phosphate group, pentose sugar, and nitrogenous base. You also learn that there are 2 types of pentose sugar used in forming monomer of the nucleic acid. One is ribose, the other one is deoxyribose. And ribose is used by the RNA and deoxyribose is used by the DNA. And then you learn about how to classify nitrogenous base. It is by purines and pyrimidines. And these are the types of nitrogenous base classified in pyrimidine, which is CTU, and A and G classified as purines. Okay. Other than that, you also learn that base A will always bind to T, and C will always bind to G. Okay, so in RNA there is no T, but we always use U. So I think that's all that we learned in macromolecules. Thank you.